Thank you, Anne. And it's my pleasure to introduce our keynote speaker for this morning. Dr. Terence Sanger is a child neurologist who is specializing in movement disorders, and he is also a computational neuroscientist. He did his pediatric training at LA County Hospital and then did pediatric neurology at Boston Children's Hospital. And now he is at California again, at Irvine. He is the vice chair of research in pediatrics and the chief scientific officer at Children's Hospital of Orange County. He has done work on high-tech studies, particularly focusing on movement disorders, which is the topic of his talk this morning. I will introduce him, and please welcome Dr. Terence Sanger.
Great. Well, thank you very much. It's a pleasure to be here, and I really appreciate the invitation to speak. This is an interesting topic, and the title is probably somewhat unusual for grand rounds. As Dr. Rizzo mentioned, my background is mixed between different things. I am a child neurologist, so I deal with brains, which are basically sort of electrochemical devices for information processing. And so, but my PhD is in electrical engineering and computer science. And then the idea is, can you use the techniques from electrical engineering to try to understand how brains work, and actually, much more importantly, how brains break? And I do like to tell my students, and at moments of despair and hopelessness, that it's much easier to figure out how something breaks than how it works. A lesson I learned from a Ford Mustang I used to own, which had the most amazing set of tubes and wires and things under the hood. And if you opened the hood, occasionally some tube would have come loose from some vent somewhere. And I didn't know what the tube did, but I did know that if I reconnected it, the thing would start working again. And so I think a lot of child neurology is like that. We don't really understand how brains work, and someday maybe we will, but hopefully not in my lifetime, or I'll be out of a job. So I'm going to talk about movement. Oh, disclosures. I wish I had financial interest to disclose, but I don't. So. And everything's off-label, but this is pediatrics, so you knew that anyway. The research I'm going to talk about comes from a new technique that I and my friends who are neurosurgeons invented originally at Children's Hospital Los Angeles, and have now brought down to Children's Hospital of Orange County. And this is a technique. Deep brain stimulation has been used extensively in adults, mostly for Parkinson's disease, and to some extent for Parkinson's disease and essential tremor were the true triumphs, and there's tremendous success, and there are hundreds of thousands, perhaps we're up into the millions of cases of deep brain stimulation being used for this. And deep brain stimulation, for those not familiar with this, involves putting wires into deep areas of the brain, typically basal ganglia or thalamus, and hooking these up to a pacemaker in the chest. And if you do that, it's called stimulation. It's modulating the signals. It's probably blocking some signals, accentuating others, changing how things go through an area. And when you do deep brain stimulation, there are just one wire on each side, and each wire has 4 contacts. You've got basically 8 places in the brain that you can stimulate. And if you realize that the brain has approximately 100 billion neurons, and we're stimulating at 8 of them, the analogy I use for that is it's like trying to repair a modern computer using a hairdryer. We really have a significant lack of specificity. But that said, it's very clear that where you point the hairdryer matters. So that you need to be very precise about where you put these leads. And there's been tremendous amount of experience in the adult world about finding the exact locations. And they can look at data from hundreds of patients with Parkinson's disease and find the exact locations. Now bring this to children. So now the movement disorder that occurs most commonly in children that I would see is dystonia. Depending on how you calculate what you consider to be a movement disorder, probably the most common one is tics, followed closely by spasticity. But dystonia, which is a distortion of voluntary movement. There's lots of definitions for this, and I'd worked on groups that had tried to define it. But I think my working definition of it is that you try to activate one muscle and you get 6 instead. It distorts voluntary movement, but it can distort anything. It can distort the voluntary attempt to hold still, to not fall off the examining table, to hold your head up. certainly to speak, to eat, to move, to walk. And so, of course, can come in very mild flavors and then can be very, very severe. And the problem with dystonia, particularly in children, is that it has many different origins. So that dystonia can come from, the most common one is various hypoxic ischemic injuries around the time of term birth would probably be the most common one. But there's certainly, we're talking about metabolic diseases. I like to say that, the basal ganglia are sort of the sentinel pigeons of the body. If anything goes wrong anywhere, sooner or later you end up with a movement disorder. And there's a lot of reasons well studied for which this area is particularly sensitive to a variety of metabolic diseases. So you can imagine that between metabolic diseases and which, whether you want to consider those to be inherited or acquired or a little mixture of both, combined with purely acquired injuries, hypoxic ischemic, trauma, infectious, vasculitic, there's a large category of diseases. And one of the things that we've learned is that dystonia can have origins in many different parts of the brain. It's not just the different etiologies. So we've seen people where certainly basal ganglia can be a cause, but thalamus can be a cause, peripheral nerve can be a cause. And more recently, there's tremendous excitement about the emerging strong data to support cerebellum as a cause. And I'm not going to talk too much about that, but what it does say is that if you have all these different causes, and in terms of anatomic location and etiology, it's pretty unlikely that putting wires in the same place for every child is going to work. And that's in fact what was seen worldwide, which is that if you try to do deep brain stimulation for children with dystonia, you only have a success rate of about 50%. And how you define success is variable. Success meaning that you have, I sort of define it as enough improvement that if you ask the parents, they do it again. But there are some scales that we use to look at outcomes of this. And so as an electrical engineer, I said, well, what I really need is to find the right spot. And what do we do in electrical engineering? We apply voltages and measure currents. And ideally, you would have some way of probing these things. And that led to the technique which I'm showing here. And this happened serendipitously. The story is that we had a young girl, she was 7 or 8 years old, very, very severe dystonia. And one of the things that happens in dystonia is because of all of the muscle contractions that are happening all the time, these children have tremendous metabolic needs. And at some point you can't feed them enough. Dystonia kills you essentially through starvation. And then the secondary consequences of that, as we know, you end up with immune failure and various infectious processes. And she was very much in sort of the end stages of this. And we tried to do deep brain stimulation on her. And the typical way of doing deep brain stimulation is that you, because you still have to find the right spot. It's still hard. Stereotactic neurosurgery is not a simple procedure. You're trying to get these wires 10, 12 centimeters into deep areas of the brain. It's easy to miss. You have to be within about a millimeter of your target to have any kind of efficacy at all. So the way that this is done in adults is that you have a whole stereotactic setup in the operating room. You wake the patient up during surgery, and during that time you will record. You drop depth electrodes, micro electrodes, record from the brain areas, listen, because we know what these brain areas sound like, and make sure we're in the right spot, put in a stimulating electrode, which will be the permanent electrode, and turn it on with a patient awake, and you look for side effects. And in some cases, you can look for beneficial effects. So now imagine a 5-year-old with their head bolted into a frame in an operating room, awake with dystonia. I mean, Parkinson's is perfect. They don't move. It's great to be in an operating room on an operating table with somebody who doesn't move. Dystonia is the other way around. You wake them up and you scare them. They start twisting and moving. And you can imagine the anesthesiologist sitting there with their finger on the propofol, trying to, and we're all, this is not a good situation. And this child was worse because to wake somebody up, you need to use an oral airway. You cannot have endotracheal intubation in an awake child. And she could not tolerate that. And so we invented this procedure for her. And when we did this the first time, the result was so successful that we never went back. It wasn't that it was what happened. And what we do is, instead of doing all this in the operating room, we put depth electrodes, the same things that are used for epilepsy, into all of the areas of the brain that we think we might want to hit. And then typically it will be 10 different areas, 5 on each side. But with the same electrode, we can hit multiple brain areas. So it tends to be about 8 or 9 areas on each side. Each of these electrodes has multiple contacts, so we can hit different areas of the brain. Child is under general anesthesia, endotracheal intubation, wakes up, and then we bring them to what we call a neuromodulation monitoring unit, but it's an epilepsy monitoring unit. We do this in epilepsy all the time. It's a known procedure, except that now we're doing it for movement disorders. And now the kid is awake in bed with the parents present, and we can get them out of bed and we can have them eating, and if they can talk, they can reach for things, they can walk. We have the parents bring in whatever it is they're trying to work on. And at the end, and we keep them for a week, and we measure, we stimulate at all of these areas, and we record from all of these areas, and we try to figure out what's best. And at the end of that week, the decision is not made by the doctors, it's made by the parents. And this, I think, may be the single most important aspect of this, is that the parents are there. The parents are not in the operating room. They can't make that decision when you're doing this under general anesthesia or under endotracheal intubation with the awake child in a head frame, but they can when they're in the hospital room. So this is actually pictures of that first child and the AP view of her head with 10 temporary wires in there during off the CT scan, and you can see on the second image there what it looks like in the operating room with these wires coming out of someone's head. It's dramatic. On the other hand, it's not unusual. We've been doing this in epilepsy for quite some time, and one of the things, on the disclaimer slide, whether the things are off label or not, in fact, this is not off label. It turns out everything is actually labeled for this purpose. I'm actually talking to the FDA about this. I was surprised to learn that. So this is the process that we do now. We use a robot-guided stereotaxy for this. So that's the big disc there, of course, is an intraoperative CT scanner. The thing, the red and white thing under the clear drapes, is a ROSA robot. There's two surgeons that work with me, Dr. Mark Leiker and Dr. Joffrey Olaya, and they have been obviously instrumental in the three of us developing this technology. And what happens is that there's many aspects to the surgery, but in the end, there's nothing that surprising about the surgery. There's nothing that wild about the surgery. What is wild is what happens later. So this is now a reconstruction of where some of the leads go. The rotating image there is showing typically a child with, in this case, our typical 5 leads, and each one of these leads has 6 contacts that we can stimulate through on each side. So we have 60 stimulation points. Each one also has 10 separate contacts that we can record through, which are higher impedance contacts. And we can use those to look at the activity in these brain areas. And I'll show you some of that. And that's the data sets that I'm going to be talking about today. But the other thing that's very exciting about this is these surgeons are very good at this. And it turns out, and I like to say that whoever designed the brain wanted us to do this because they lined up all the right structures. So these things actually are in line, and it looks a little bit like a shish kebab, but the structures we want to hit are there. And in fact, it was very funny because we're sitting in the operating room one time, and when I was trying to do this, and I wanted to align into the VIM nucleus of the thalamus, and I wanted something into the pedunculopontine nucleus, which has been shown in adults to be very good for sort of midline axial tone problems. And the surgeon said, well, I can't do that because the 2 wires will lie right on top of each other. I'm like, wait a minute, what if we used one, right? And in fact, that turned out to be the case. Somebody designed the system with the pedunculopontine nucleus lined up with the VIM nucleus of the thalamus, and I'm very grateful for that. There is other areas where we have the same thing with the ventral oralis nuclei of the thalamus line up very nicely with the subthalamic nucleus and the substantia nigra reticulata, so that we can hit some of these. There's some things obviously that we can't hit, but I take that as a message as well. So that is the sort of the structures that we do, and then we get data out of this. And one of the things that anybody who studies neurology dreads the pictures of the basal ganglia with the boxes and the arrows and the lines, and neurologists have suffered through this for years. And there's a rough idea that much of the connectivity within basal ganglia is mediated by GABAergic synapses, which are inhibitory. And so you have something inhibits something else, something inhibits something else, something. You're trying to count up all the inhibitory things and figure out how many negatives make a positive. And the idea is that if you put all this stuff together, this area goes up and inhibits that one, which goes down, which disinhibits that one, which goes up. And so there's all of these predictions, all of which we've known for a long time are wrong. And what we've seen here is exactly that same phenomenon. Everything that we thought about this inhibitory and excitatory system turns out to be wrong. And when you think about it, that sort of makes sense, because there's no point in inhibiting something that's off, right? So you only want to inhibit things that are on. So it's not likely. So if you have one brain area inhibiting another, it's not that this is going to be on and that's going to be off. It's they're both going to be on, but this one's sculpting the activity in that using these inhibitory synapses. It's and in fact, the basal ganglia, historically, it's very interesting, because we had always thought, we meaning back in the 1950s and 60s, that information was transmitted on excitatory synapses and inhibitory synapses were purely modulating. And there are two major areas in the brain where inhibitory systems seem to carry the information. Basal ganglia is one, and cerebellum is the other. So there's something about movement control where inhibition seems to be very, very important in transmission of information. But what we see is during movement, whether it's voluntary movement with or without dystonia, it could be a dystonic spasm, everything turns on when you move, everything turns off when you don't. This is energetically efficient. It's a good idea. It's just like your Tesla, just like any car with a, with automatic starters. You come to a stoplight, your engine shuts down. That's a good idea, right? You're not moving. Why use gas? So it's the same kind of thing. The brain tends to shut down all of these areas when you're not moving, and they all go on. So the problem then has to be in the patterns. When there's a problem, it's not about, is this area firing too much or too little? It's what is in there. Now, that doesn't necessarily apply always. Parkinson's disease, there are definitely areas that are firing too little and definitely areas that are firing too much. So Parkinson's may be the example of a disease where things are too hot or too cold. But I think dystonia is much more a disease of abnormal patterns, and I'm certainly not the first person to say that. This is an example, and this is where we're getting. So one of the big conundrums in all of this, and because so much of the field has been driven by this idea that an area is on or an area is off, or it's on too much or it's off too little or something, much of it has been driven by what we call these rate models, the actual rate. And people have started to look at patterns. And again, this has become very important throughout adult movement disorders. The pattern that causes dystonia is not well known, but what we've started to see, I've now done, we have recording data on 45 patients that we've taken through this process. And the recording data tends to show the same thing over and over again. And in fact, we're just pulling this stuff together now, so you may be one of the first audiences to hear this. But what you see on the top here is that's a time tracing. So time is running from left to right, and what's running vertically on the top part with all those colors, every single row is a different neuron. And we're pulling out the spikes from these neurons as the neurons fire. And at the bottom, we have the EMG. And this is a patient who was having spontaneous dystonic spasms. So they're lying there in bed and the spasms are starting. Now, I don't actually think spontaneous dystonic spasms are a thing. I think what happens is that this is probably triggered by emotion, discomfort. It could be triggered by attempts at movement, could be triggered by attempts not to move. But there's usually some kind of a trigger. But whatever's happening, what you see here that's characteristic is this bursting activity. You see this sort of non-rhythmic bursting that's happening across all brain areas. And if you try to say, where is it coming from? There's no one place. It's not propagating. It's not starting at one place. You know, you'd think like in epilepsy, you can see a focus and it starts at one spot and it will sort of spread. And we've looked very carefully for that, and it doesn't look like that at all. So everything goes on at once. Everything goes off at once, and so that's what we call a network disorder. The instability is an instability in the overall network. You know, if I take this microphone I put in front of a loudspeaker, it starts to screech. That screeching happens because of the feedback. But which came first, the microphone or the speaker? And the answer is both. It is the phenomenon happens simultaneously at the moment that the microphone is put in front of the speaker. And I think it's exactly the same thing here. This is feedback instability. It's a well-known phenomenon in electrical engineering and control theory, which brings me back to my background in control theory and signal processing, where we see this stuff all the time. We don't see it exactly like this. Robots tend to explode. Fortunately, children don't explode. They just become dystonic. It's a very similar phenomenon too if you just turn the volume up too much on these things. And the thing here that's unusual is this asynchronous bursting. Now, there's another interesting phenomenon that I think is important about this sort of asynchronous aspect, and this is one of the things that has bothered me for a long time, and I don't have an answer to this. Deep brain stimulation. This is a very delicate area of the brain, right? We know that very small strokes, very small injuries to this area of the brain can cause tremendous disorders of movement. So why can I put a wire into it, put 5 volts and 2 or 3 milliamps on that wire, and most of the time what happens is nothing. Most of the time people completely tolerate this. It's bizarre. We should be causing seizures. We having people should be curling up in a ball. It just doesn't happen. And in fact, it's so safe to do this that the modern devices for this, when we do the permanent implants in the chest, these are controllable from the outside. We give the parents the ability to turn the voltages up and down because sometimes it makes kids better. But we know it's never going to cause a seizure. It's never going to actually to make that, well, it can make the dystonia worse, but never in a catastrophic way. And it's surprising because, you know, we're talking about neurons that are operating in the millivolt range, right? The depolarization potentials are 60 to 90 millivolts. And we could be putting in 4 or 5 volts into these areas, right? So why doesn't it cause a problem? And I had a little bit of a clue from that, looking at recordings in epilepsy. One of the interesting things that happens, and again, I talk about epilepsy as if it's one thing. Obviously, it's many, many different things. But if you look at cortical recordings in epilepsy, and you look at the propagation of waves of epileptic activity across cortex, these will propagate for a while, but they look a lot like the activity I'm showing here. It looks it's sort of a wave front, but it's kind of random and asynchronous. What precedes the end of a seizure is synchronicity. As soon as things start lining up, as soon as the activity of the neuron starts being completely synchronous, it shuts down. And so I think that the brain is actually very good at shutting down things that it doesn't like. There's a whole sort of police mechanism for epilepsy in there that may, and again, I'm not an epileptologist, but so this is a movement disorders view of epilepsy. So my apologies to the epileptologists out there, but that there's a police mechanism that says that things that are firing completely synchronously across a local area, that's not normal and it's not conveying information, and we're going to shut you down. And that those mechanisms come in and may be responsible for things like spreading depression. There's a whole inhibitory mechanisms in the cortex that do this. So I think that the survival of an epileptic wave depends on lack of synchronicity. So it depends on grouped firing, but that is not synchronous. Now, again, I'm not going to make a claim about epilepsy. There's plenty of people studying that. But I think that in this case, that may be exactly what we're seeing here. The propagation of a dystonic signal depends upon it not being synchronous, depends upon all of these bursting activity. But it's not a line. It's a time, it's a region, and you can see that happening across these areas, and you see it correlating with the muscle activity. Down below in the bottom, I show the IM nucleus of the thalamus and the CM and CL nuclei of the thalamus. Both of these are output channels from cerebellum, and in this case, we thought that cerebellum might have been an origin of or at least a contributor to this child's dystonia. And in this case, you see that the summed bursting activity, so the purple and the green shows you the bursting activity, and overlaid on that is the summed EMG, the electromyography from the muscles, showing that these things are highly correlating with each other. Presumably, these bursts eventually make it from the deep nuclei in the thalamus up into the motor cortex, where they propagate down to the muscles. So, but again, so I think there's a very specific quality to this, which may be the reason why deep brain stimulation can treat but not cause dystonia. And it's because deep brain stimulation to the brain is I've depolarized a whole bunch of neurons at exactly the same time. And not only exactly the same time, it's completely rhythmic. So that the brain can predict when the next pulse is going to come and shut the thing down. But that shutting down process may actually be important. So it may be that the reason why the deep brain stimulation looks a little bit like a lesion, and before we had the pacemakers and the stimulation, people made lesions in these areas, then maybe the reason why it looks like that is not because it's stimulating, but it's stimulating in a way that the brain recognizes as alien and shuts down. And by shutting down that region, it behaves a little bit like a lesion. Deep brain stimulation, however, is not exactly a lesion, because when we put our electrodes in and stimulate, the stimulation itself propagates through the neural pathways and leads to activity in a whole pattern of areas throughout the brain. So that pattern means that we're not only blocking activity or modulating activity at one spot, we modulate that activity over a whole region. Now, we don't understand how to control that. We don't know what we want to do with that, but I think that is one of the reasons why the stimulation is fundamentally very different because than say making a lesion in one spot. Okay. There is, during those bursts, excessive spike synchronization, as is seen in epilepsy, and we do see this. It's just not everywhere. This excessive spike synchronization can happen. There's some probability if you have a bunch of neurons and they're just firing more, there's more of a chance that two of them will fire at the same time. You know, the more people in a room, there's a higher and higher chance that two of you have exactly the same birthday. I think with the number of people in this room, there's probably at least 20 or 30 of you who have the same birthday. So there's a rough chance of that happening just from that. But you can calculate the excess chance of synchronous synchronous firing, and it's much higher than you'd expect. And in fact, the synchronicity here is incredibly precise. What we're showing here, for instance, in these different traces, is we look at one spike and we average another channel based on one spike. So I take a spike from one channel and I use that to synchronize, to time average another channel. And on the second channel, we see a spike that is, and these things can be millimeters apart. Millimeter in the brain is huge, right? This is a very, very big difference. So it's not on the same electrode. These are different contacts, different electrodes. We can see this. So that there is millisecond level precision of spike timing. And you know, how can that possibly happen? Even if these two neurons were connected to each other, I don't think that's a possibility because it'll take a millisecond or so just for the signal to propagate from one to the other. So I think what this represents is upstream synchrony. So people who work in the neuromuscular field are used to the idea of what's called grouped atrophy and large motor units. And so if you have the cells in your spinal cord control your muscles, and they will send different signals down to different muscle fibers. If you start killing the cells in the spinal cord for whatever reason, you've got some neuromuscular problem. The remaining surviving axons down to the muscle will sprout, and they will start to contact multiple muscle fibers from the same incoming nerve fiber. Now those muscle fibers all fire at the same time, and you will get relatively large motor units that are firing very synchronously in abnormal recruitment patterns. I think very much the same thing may be happening here. So upstream, you kill an area in, say, the striatum, or you injure it. There's fewer neurons there. What happens downstream from that? The surviving neurons will innervate a larger region of the downstream neurons, but they're now getting simultaneous input. Now you get the simultaneous input. You have this synchronization that's happening over all of these, and that synchronous activity is highly abnormal. It's not only it's abnormal, it's now going to cause problems, because now downstream from that, 2 synapses further down, those neurons are getting bombarded with simultaneous input, which is not a good thing. This simultaneous input is too much. They're not expecting that, and we do know that simultaneous input to neurons can not only lead to abnormal information processing, it can actually lead to cell death, just because of the excessive calcium release and the excessive metabolic demands on the cells downstream. So there may be a cascade of problems that happen due to the synchronicity, and again, all of these things are in a loop, so they feed around, and now you have that. So it's a hypothesis, but again, there's evidence of significantly abnormal processing, and we can see this excessive spike synchronization across multiple areas, usually within an area. Although in this case, you see it actually between areas. And this is what I was saying. There's no time. The time lag here is 1 millisecond. This is a cross-correlogram. The green lines are pointing to areas where, say, a lead in one part of the globus pallidus correlates with a different globus pallidus, but the centromedian centrolateral nuclei of the thalamus are also correlating with very, very low latency. Again, 1 millisecond latency, which is not, so that's shorter than the propagation time with the globus pallidus, to which they project. So there's a connection between these. But again, there's what I think is happening in this case is that it's this simultaneous bursting. It's not, it's the microphone and the speaker problem, or the chicken and the egg. It's you don't have there's nothing came first. the whole thing happen. Chickens and eggs happen simultaneously. Screeching from a microphone and a speaker happens simultaneously. That's what's happening here. Okay, how do we do with all of this? So by doing all of this, I'm looking at all of these areas, and I can, from this, we can now decide, okay, where do we put the leads? I have a child in the hospital for a week. We get all this data. We look at the various areas. We look where there's normal firing and abnormal firing, and we can test stimulate and see what happens. The upper left is showing preoperative. Preoperative is sort of in this particular case, it's a misnomer. She already had deep brain stimulation leads in place. She'd had multiple complications. They'd had to be removed, replaced. And at this point, DYT1 is a genetic disorder that's known to respond very, very well to deep brain stimulation. And the fact that she has deep brain stimulation leads in place and is not responding was due to many complications. So we removed those leads and ended up trying to replace them. On the lower left, I'm showing what we were testing in the neuromodulation monitoring unit. This is showing a child. So she has 10 wires in her head and 160 channels. My hardworking graduate students are pushing along the equipment rack, which is recording from this, and the physical therapists and the nurses are all managing the stimulation. And we can have her walking down the hallway as we test stimulation and see what makes a difference. I mean, this is, just to put this in context, this has never been done in humans, right? It's hard to do this in animals, right? This is, from a primate research standpoint, we're learning a lot about walking by doing this. But the good news is that afterwards we could find good spots, which are different from where her existing leads were. And we tend to put 4 wires in everybody because we can, and it almost always helps. In her case, we have wires in both basal ganglia and thalamus, and made a huge difference. And she was able to return to most of her normal activities. It's a difficult programming, and there's the follow-up to this is not straightforward. This is a child with pantothenate kinase, also a disorder that's known to respond to deep brain stimulation. It's a progressive disorder, although probably not as rapidly progressive as we used to think. The dystonia makes these kids appear much worse than they actually are. The cognitive function in these children is probably relatively preserved throughout much of the phases of this disorder. His problem was that he's an adult, he's 18 years old. He refused to get a G-tube, and he insisted on eating himself, which required his mother to perform the Heimlich maneuver several times because he would severely choke. And the problem with deep brain stimulation is we've always said it doesn't have much of an effect on speech and swallowing, except that this child was going to die. Well, we could have gotten him a G-tube, I suppose, but he was absolutely refusing it. Where his targets ended up being in the globus pallidus, which is the known target for pantothenate kinase, and the pedunculopontine nucleus in the brainstem. He's one of the first kids that we went into brainstem. We did this based on data from the adult world and based on understanding the neuroanatomy. And the most miraculous thing about this is as he's sitting there in the NMU, and imagine these are not things you could do in an operating room, right? We needed to see him eat. And so we told his mom, bring in some food and come back the next morning. And she didn't just bring in food. I mean, there was like piles of food, his steak and burritos and stuff, right? And she's like, well, he's eating for the first time in 3 years. And you've got these temporary wires in for a week. I'm going to fatten him up. and it was I mean, he really did gain like 15 pounds that week, right? And because we have to take the temporary wires out before we put the permanent ones in, and she knew this, and every time he comes in, this is clinic. This is what clinic looks like. I mean, he's just constantly eating, but he's just so proud of this. And it's, programming pedunculopontine nucleus is not for the faint of heart. The brain is very plastic. It cares deeply about restoring whatever it perceives as homeostasis, and homeostasis does not include having stimulation in your pedunculopontine nucleus. So the brain fights us. And the programming afterwards, these are programmed through telemetry, and we can we make changes, but in this case, it's a continuous fight. So we do one thing, the brain compensates. We do something else, the brain compensates. We have to be reprogramming in this for the brainstem targets about every 3 to 4 days. But eventually, you find an equilibrium. And when you get to that equilibrium, you do very well. We knew with this had worked really quite well for him. He managed to break the wires and went back to his previous stage. So we knew it was working. We've replaced the wires and now he's recovered again. So he managed to do the on-off research protocol for us to prove that this was not placebo. We knew that, but it's and I wish he hadn't broken the wires. But this is another case, GPI and VIM. VIM nucleus is the thalamic relay for much of the cerebellar output. And again, I think cerebellum is to blame and deserves a lot of blame for a lot of dystonia, whether it's because of an abnormality in the cerebellum or whether it's because it's transmitting abnormal signals from the periphery or from brainstem. I don't know. But certainly by blocking or modulating its output, we often do quite well. And this is the kind of thing where he had the severe, this hyperkinetic form of dystonia. Difficult to distinguish from chorea, but this is a hyperkinetic dystonia. And we were able to get a lot of the hyperkinetic components under control. What I'm demonstrating here, so preoperative is on the left and postoperative is on the right. It's not normal, right? Well, I can't, these kids, there's dead areas of brain, right? I'm not fixing that. And again, this is the hairdryer. All I can do is cool it down or warm it up, right? I can't do that much. And in fact, different components of this get better or worse. So in kernicterus, as with many of these kids, there's the hyperkinetic form of dystonia where you have excess movement. And there's the hypertonic form of dystonia where you become very stiff. I used to think those were two sides of the same coin, that the only difference is that, if I contract my biceps and triceps at the same time, I get stiff. If they're not quite at the same time, I move. In fact, I think those are probably different. They're probably actually very different phenomena. We call them both dystonia because they share the property of abnormal muscle activity triggered by attempts at movement. But they do seem to come from different parts of the brain, or at least treatment is very different. So stimulation in thalamus, we're getting almost 100% success rate with hyperkinetic dystonia. So to the point where if a child has hyperkinetic dystonia and we put the stimulating leads in and we don't fix it, we'll usually move the leads. Because we really do think thalamus, and it's not always VIM thalamus, but it's that region. We think thalamus really is the origin of this. Hypertonic components of dystonia respond to globus pallidus, and really we don't get the same kind of benefit. We get about a 20% reduction maybe in most of the kids. Now that can be good. 20% reduction can allow you to sit in your wheelchair or your car seat, or maybe stand and use a walker. But it's still, it's certainly, I don't think we understand hypertonic dystonia and its treatment as well as hyperkinetic. But the fact that there's a dissociation in these brain areas and suggests that in fact these are different phenomena that just happen to coexist in the category of etiologies that cause damage to basal ganglia and thalamus. So in other words, just in his case, kernicterus, the bilirubin toxicity presumably affects multiple areas of this, and the multiple areas that are affected may include the areas that affect both hypertonic and hyperkinetic components. That's also going to be true for hypoxic ischemic injury, for certain other toxic injuries, metabolic and toxic injuries that tend to affect these. In the DYT1 dystonia actually doesn't have as much of a hyperkinetic component. It can look a little hyperkinetic, but if you watch them very carefully, what happens is you see fixed hypertonic postures that are inserted at sort of these actually often very predictable intervals, but they're not nearly as hyperkinetic. The first girl I showed you where we did the reoperation, one of the things that drove us to that is she was hyperkinetic with DYT1 dystonia. So that said that her system was operating very, very differently. And that's why we ended up in her going into both pallidum and thalamus. Okay, here's the usual pictures of this, and I'm not going to go through this, but there's many more. You could draw arrows in all directions. Everything connects to everything, and we don't really understand all of this. But I think one of the things that I do want to point out here, the most right-sided oval where it says VOA, VOP, and VIM. Just want to point out VIM is the output nucleus of the cerebellum, and VOA, VOP is the output nucleus from the basal ganglia. We can roughly divide the thalamus into sensory and motor components, with the sensory areas being a little bit more posterior and the motor being a little bit more anterior. There's probably some crosstalk between those, but not that much. And I think that the reason this is important is to recognize that sometimes we're affecting these sensory loops, sometimes we're affecting the motor loops. and with the stimulation, because both VOA and VO and VIM are targets, as is GPI, so that this comes back to this idea that dystonia can have many different origins, many different anatomic origins. Anywhere there's a loop, all electrical engineers know this. Anywhere there's a loop, you can have instability. Anytime there's feedback, it's possible to turn the gain up too much and get oscillation. There's loops everywhere. There's loops through the periphery. There's loops through the brainstem and cerebellum. There's loops within the brainstem. There's loops from cerebral cortex through thalamus. There's loops through basal ganglia. These loops are everywhere. Why the brain isn't continuously oscillating is the true miracle of all of this. And like I say, I think a lot of these origins of this may be sort of failures of policing mechanisms. So now one of the things that comes out of this is this gives us the opportunity to look at how information is coded within the basal ganglia. Now, I am never going to have a healthy child and undergo this procedure. So we will never have controls. And it is remarkable the number of, I can't resist. It's remarkable the number of feedback from study sections at NIH I get saying, you need control subjects. I'm like, okay, you do have a child who's volunteering. But this is a common problem for this kind of thing, right? Which is we don't have the controls. On the other hand, it's not a problem because I don't care about healthy children. I mean, I do care about healthy children, but I don't, but as a neurologist, right there, a healthy child shows up in my office, it's not going to happen, and that's not my job, right? So that's fun on the playground, but it's so in some ways this is the data I want. I want the data from the unhealthy kids, but I need a range because I need to know what am I trying to accomplish, right? The whole goal is if you see is it abnormal or is it not abnormal, right? And that can be hard to figure out. And the closest thing to a healthy person that we had was a child with severe Tourette syndrome. And a young adult, actually, very, very severe Tourette syndrome, and full, echolalia, coprolalia, just, horrible stuff. We put the electrodes into him, and we had 12 electrodes in different brain areas. And he woke up from that surgery with the temporary electrodes in his head, didn't have a single tick for 2 weeks, which was a disaster because we had to see the ticks or we couldn't figure out what area to stimulate would make him better. So we cured Tourette syndrome for 2 weeks. And you have to imagine, we were coming into the room, we're putting him under stress situations, we're giving him, Ritalin, we're doing everything we can. Couldn't get a single tick out of this kid. There's nothing, right? They started to come back after 2 weeks in the hospital. Usually we only keep these kids in for like 1 week. Started to come back enough that we could finally see them. But during those 2 weeks, the recordings are the closest to normal we're going to get, because he has normal, when he's not having a tick, the kid's motor function is normal. I don't know that his brain is normal, but his motor function was completely normal. He did very well in terms of his Tourette syndrome afterwards, although Tourette's, like peduncular punctate nucleus, Tourette's seems to have a very high plasticity rate, and we've seen in him, as with many other kids, that the effect seems to wear off, which is true in him, and we probably need to move his electrodes. But the idea is that Tourette syndrome is at least temporarily curable, although it requires putting 12 wires in your head to do it. So maybe we need a better solution for that. But along the way, we can do these recordings. Now, people who've studied cortex know that there's a lot of data from recording in cortex. I can't say that we fully understand what cortex does, but if you look at motor cortex and premotor regions of cortex, there's a very clear relationship between the firing of individual neurons and, say, the direction of arm movement, and in some cases the particular muscles that are used or the direction of force that's applied. In basal ganglia, there isn't. We just don't see it. And the kind of things that we see, and so this clear tuning for direction, I mean, in cortex is beautiful, and Apostolos Georgopoulos and others have shown this for many years. Decoding movement from cortex, some of you have probably seen the television, people put chips into somebody's head, and you can use that to control a mouse or a wheelchair or a robot. Monkeys can learn to do this. I don't think this is the future of human-computer interaction, but certainly we know how to read that out. From basal ganglia, almost impossible. Is it on or is it off? Is about as good as you can get. And that's kind of what we're seeing, although what we do tend to see is temporal patterns. So we tend to see neurons that will fire before movement, or before the movement starts, before the movement ends. So this is a case in the GPI comparing the VOA, VOP. The vertical line in the middle is the start of movement. The red sort of S-shaped line to the right is the time at which movement ends. So we've sorted these movements by movement time. And you can see in GPI, there's a this is a bunch of different cells, and you can see that many of the cells will respond immediately after the onset of movement, and maybe around the time of the end of movement, but not during the movement. Now, this is different, right? If you think about that, these cells can't be driving the movement, right? They're just starting. It's just the beginning and the end. They're telling you about changes of movement. They're not. A cell in motor cortex is actually driving the movement, right? It has to turn that muscle on during the whole movement, right? But here, that muscle is on during the whole movement, but here you have things that are just sort of, when does it start? When does it stop? Now, in thalamus on the right, you see things that are a little bit more active between the start and end of movement. But again, it's different. Depending on which cell we look at, we can get very, very different types of responses from these. Most neurons don't seem to have anything to do with task timing. Now, I do point out again to my students in moments of when things don't seem to be working, is that the basal ganglia don't just care about what we're doing. Just because I have a child doing finger to nose or something, or they're tapping buttons or doing a go, no go task or something, the basal ganglia are maybe paying attention to that with, part of their basal ganglia brain, but the brain's doing a lot of other things. The kid's hungry, he's thinking about lunch, mom's over there, what's on the TV, they're trying to sit up in bed, the IV's making them uncomfortable. I mean, there are so many things going on in these children's head at any given point. So we have to sort of pull out which part of it has anything to do with movement, which is probably a minority. And this is true in all experiments. The people doing monkey experiments, you can bet that monkey might be reaching for something, but what's really thinking is, get me the heck out of here, right? So, and it's trying to do that, right? It's thinking, if I reach for that, can I get the experimenter's tie or something, right? I mean, it's very so. Okay, so, but when we look at this, it leads to this possibility: could the patterns actually be random? And this is an interesting problem. There's I'm going to come back to this. A fascinating fact of humans, which is that we can do amazing memorization tricks. This was a study done by Lionel Standing quite a while back in the 70s, and he showed people 10,000 images, one per second, just one. You just got a chance to see each one once, and this happened over 3 or 4 days. And then a week later, he'd show you a bunch of random images and say, "Have you seen this one before?" Now, people weren't perfect, but they were much better than chance. So imagine that 10,000 images, you see each one once, and not just better than chance. Like if there was one image, I think they said, where it showed a piece of bread inside a bread box. I'm not sure I know what a bread box is, but anyway, he had pieces of bread inside some sort of a box, and they showed another picture that showed the piece of bread half out of the box, and people said, I think I've seen that before, but something's wrong. So the idea, one-shot learning. So how do you do one-shot learning? One of the best ways to do that is what we call hash codes, because computers can do one-shot learning, right? And what you can do is you can take these very complex images and you can say, okay, I'm just going to pull out a few pixels from each one. I'm just going to remember those. And I can't reconstruct the image, but I can say if it's an exact match, did I see it before? But these hash codes look random. This is encryption. This is what we do with our passwords, right? You don't have to, when you type a password into the computer doesn't regenerate that password. The computer takes that password, changes it into some crazy hash code, and compares that hash code. If somebody looks at the hash code, they can't regenerate your password from it. That's the whole point, right? And so we do this in computers all the time. And I think the brain may actually be doing this, which is bad news for a lot of us, including Elon Musk, because the point is it means that you may not be able to decode lots of things from your brain, right? It may actually be encrypted. That said, we have to try, because this is trying to understand what's going on inside basal ganglia is going to be very important if we're going to try to actually modify these patterns. This is just an example of what sort of random dithering looks like, but the level of the problem that we have to solve. On the left is an original image of a child running. In the middle is a dithered image. And this is maybe what the brain actually sees. Dither just means that every pixel is on or off with a probability equal to the intensity of the image. And that's kind of how neurons do it, right? They'll fire more for bright areas and less for dark areas. Okay, fine. But now I'm just going to take all of those neurons, I'm just going to mess them up, right? Because in order to produce this image, I need to know which one is the upper left and which one's next to it and which one's next to it. But inside the brain is just a bunch of wires coming in, right? Now the brain knows, but I don't know, right? And once you're in, if you have the somatotopy that exists in primary sensory areas or some of the primary motor areas, that's fine. We know, the knee bone's connected to the ankle bone, right? I mean, these things are close to each other due to the somatotopy. But when we look at basal ganglia, this is mixing information from sensory and motor areas, in fact, from both sides of the brain. So now you take that image on in the middle, and we just randomly scramble the pixels, and now you can't see a thing. And our job is to unscramble the right image to produce the left one. So good luck, right? And I don't know how to do that. There's another problem that happens, which is that a lot of people try to understand what's going on in the brain by saying, can I approximate something from it? So if I can take an area of the brain and I can approximate, say, the muscle activity from it, then the muscle activity must be what that area of the brain is doing. Remember, we don't know what the basal ganglia are doing. So we'd like to use this kind of technique, and this has been used with some success in cortex. The problem is hash codes, this random coding, is actually a universal approximator. You can approximate anything. So on the left, I'm showing approximating muscle. So the red line is the muscle activity. The blue line is the approximation of it calculated off basal ganglia. And I show at the top and the bottom on the left that you can get different values of approximation just based on how you do the approximation, just based on the filtering. But on the right, I take something which has nothing to do with the brain, which is a square wave. And I say, let's just try and approximate a square wave using the same technique from actual brain activity. And look, I can do it. It's beautiful. It works, right? So does that mean the brain was approximating a square wave? No, it just means that we have this incredibly mathematically powerful thing. The brain could have approximated a square wave if it wanted to, but the fact that we can do it doesn't mean that that's what the brain is doing. So one of the things, so now back to the original topic of my talk in the last 7 minutes or so here, which is the idea is that if this is really encrypted, then let's be code breakers. Let's think about the principles of cryptography applied to these things. And this is very much work in progress. And how do we do cryptography? What are the things that allow you to break codes? And these are actually more ciphers than codes. A cipher is where you sort of take a letter. It's a secret decoder ring, right? You take each of the 26 letters of the alphabet, you map it into a number, and you got to figure out which is which. Codes are much more like the strong encryption that we hope is protecting our bank accounts. One of the things that people use is statistics. We know E is the most common letter in the alphabet. Find the most common thing that occurs. Let's see if it's there, right? Now, we can't do that because I don't know the alphabet. So I can't use that one, right? I don't know what the basal ganglia are trying to code. I don't have the output of this. But you can use a common statistics. Code words that happen with the same statistics may be encoding similar things. There may be common sequences, things that tend to occur after each other that we can see. And there may be coincidences, which tend to not be coincidental. I don't think anything in the brain is random. It's a machine. All of these, every neuron fires because something caused it to fire. It's just there's so many possible inputs and modulators that it looks random to us. But these are the kinds of principles of cryptography. You can try to move, and this is again coming from a bunch of cells. You can look at the original firing patterns, and you can try to group cells that tend to fire at the same time into groups using some techniques. There's multidimensional scaling techniques in mathematics. But one of the things that I think has been very helpful to me is to take inspiration from the design of the cerebellum itself. The cerebellum takes things in groups of 4. It takes approximately, but it generally takes things in groups of 4 and looks at patterns within what may be random groups of 4. And why 4? I don't know. It seems to be 4. Most animals, it's 4. So it'll take 4 neurons coming in and say, okay, if all 4 of these neurons fire, I'm going to call that a thing. And if all 4 of these neurons fire, I'm going to call that a thing. And those groups of 4 may be random, or at least random, maybe with some learning tuning. And so I'm going to use that to determine coincidences, to say, okay, maybe when this neuron fires at the same time as that neuron, it means something. Right? And I'm going to try to pull that information in. And then I'm going to try to use find a low dimensional representation. The idea being that, there's, again, I'm probably I'm recording typically 250 to 300 neurons at a time. But I think that they're probably only representing a few things in the world. Again, it may be the reaching movement of the arm. It may be the position of the child's mom. It may be whether the kid's hungry or not, but it's probably a smaller number. So if we can find a small number of variables that explain everything that's going on, maybe that would give some clue. And there's a couple of techniques that are used for that, and I'm using some very simple ones. We also do this using machine learning and some of the more modern AI techniques, but it turns out you don't need that much of that. And so we look for these common sequences. And the way that we do it is in a group of neurons that I was showing you before. So time is going left to right, and each row is a neuron. We look at little chunks of this. And what I say is, can the activity at one time predict the activity at the next time? And more importantly, can it predict that activity through a low number of what we call latent variables, a low number of explanatory variables? So if the prediction from one time to the next can be done in a low dimension, maybe that dimension means something to the brain. And again, I don't know, I'm still not relating this to anything in the outside world. This is just looking at looking at the code itself, and we can do these things. And it turns out if you do that, and then so first you say what's it coding, and then you say is it related to anything in the outside world? It does seem like it's a little bit related to the outside world. Let's see if I could show this a little bit better. This is trying to predict EMG. So the red lines going down is the muscle activity. The blue lines going up are on the left are the predicted values of the latent variables. But if I then take linear combinations of these latent variables, of the coded variables, and I try to predict the EMG from that, which is what I'm doing on the right side here, I can do pretty well. Not great, but pretty well. So the key is the information is in there, but you have to pull it out. The information is in the coincidences, it's in the patterns, it's in the sequence, but it's not in the individual cells. It's coded by the pattern, not by the individual cells. Remember, in cortex, an individual cell will have a direct correlation to EMG. In our case, there's no individual cell that will directly correlate to EMG. It's in the pattern. And I think what this is reflecting is that the basal ganglia are looking at these patterns, and these patterns become very important for control and for timing. So again, you want to start and stop a movement. A movement is not a single muscle. A movement is a pattern of muscles. It's a pattern of muscles over time. So I think being able to look at patterns of muscles over time, identify those, be very specific to them, and then control exactly that pattern becomes very critical. Now, this is just the early days of trying to do this, but this is really the first time that I or anybody has been able to get reasonable information out of basal ganglia of this nature. There's certainly lots of things you can get. Again, it does fire more with movement, and it fires less when you're not moving. We always have that. So there's and EMG, muscles will fire more when you're moving, fire less when you're not moving. There's always at that time scale, you're going to have the correlations. But the point is, can you actually predict down to the level of sort of individual muscle firings and twitches what's going on? These are not, the basal ganglia are not causing this. They're modulating the thalamus and the cortex, which are probably leading to the actual control of these muscle movements. And again, like I say, a lot of this modulation is happening through timing, through saying when do I start, when do I stop. It's a dynamic system, right? Dynamics are the control of things over time. I call this neural hash coding, and hash coding is a computer term for this sort of random encoding. But I think that may be a lot of what's going on. The nice thing about hash coding is it's a random code. In other words, how you represent your grandmother's face in your brain may be random. It's going to be different for each of us. It's random, but it's the same every time you see your grandmother. Right? And it's the same thing here. Every time you make a movement, your representation of that movement and mine is going to be very different. But mine is always the same from time to time, and yours is always the same over time. And so I think that is, and when you do that, you can learn amazing things. It gives the brain tremendous computational power. Remember, 10,000 images over 3 days, right? So imagine how quickly you can learn movements. You still have to practice movements a lot to get them exactly. But if somebody shows you something once, it's just demonstrated, it's in there forever. I mean, how many times have you said, I wish I could unsee that? Right? You can't, right? These things are in there. It's stored instantly. And this is really important for survival, because the animal that learns fastest eats the one that's learning a little bit more slowly, right? This is really very important. And so I think that these hash codes are a tremendous computational tool that our brain has that turn out to be really, really annoying for experimenters to decode. But I think using these techniques, we can decode it and then unwind this whole process, going back to the deep brain stimulation. How do we build better deep brain stimulators that aren't just hair dryers, that are actually not just more or less, but actually can change these patterns to give the children the patterns they want? There's way too many people to thank, but my lab, my coordinators, all the children, all their parents, these parents, I mean, you guys know what the pediatricians, yeah, I mean, I have no idea how they do it. And I am so grateful to them for the insights the parents give me, and for the insights that my patients give me. They tell me, they'll tell you that was harder, that was easier, this feels like this, you know, when you turn the stimulator on, feels like that. We need that. We need all of our patients and parents to be helping themselves and helping the next generation. So thank you again. I really appreciate being here.
Dr. Sanger, that was simply a mind-blowing discussion. I will open the floor for questions. I can't imagine that there aren't 100. So we'll start with Dr. Peoples back here.
So thanks again, life-changing work for these patients. So I am a neonatologist and focus on hypoxic ischemic brain injury. So a lot of your patients, but 5 to 10 years prior to you meeting them. Our kind of disease process is all about energy, right? Energy in, energy out. And when we fail to have enough energy in, and you talk about kind of the issues with feeding and nutritionally supporting these children. So neuronal firing is not a metabolically inexpensive process. And so I do wonder about whether or not you guys have seen, as you nutritionally replete these children, you're able to cool their muscles down and get better nutrition into them. Do you see that you need less modulation, that's changing as the neurons have more energy and are able to fire? Is there a post burrito kind of effect, as it were, as these babies move forward, or children?
That's a fascinating suggestion. I can't say that I have, but it's something that there's so many things happening simultaneously, and we haven't specifically looked at that variable. Nutrition is hugely important. And every child who comes into my clinic, I tell the parents that. I say, you know, nobody dies from dystonia. You die from malnutrition, and you die from pneumonia. And I'm like, this is critically important. And one of the primary reasons we often do deep brain stimulation, intrathecal backbone pumps, any intervention we're doing, is for the nutritional importance of just reducing the metabolic demands on the patient. But the effect of nutrition on growth and on brain growth and development, of course, is uncontested. This is hugely important. So over time, I think it's going to be another sort of cycle. If you allow the child to eat more, you reduce their metabolic demands during a time of brain growth and cognitive development, then they will do better over time. So I've not done those studies, but I would be very hopeful and expect that the outcomes would be strongly positive.
Hey, quick question here. I'm one of the neurosurgeons here, and I walked in a little late, so sorry about that. But I'm looking at that picture up there. I was wondering, what are all the nuclei that you're targeting? I picked up on GPI, PPN, substantia nigra, and why so many electrodes? Is each one going to a different nuclei, or are you trying to tease apart the internuclei differences that we see when we do, for example, epilepsy treatments, and we're seeing some of that changes?
Yeah, this is very similar to what you would do in epilepsy, and I think I made that comment before you got here. But just to the electrodes, because we're using the AdTech MM16C electrodes, which have 16 contacts. So there's 6 contacts at about a 5 millimeter spacing, and then there's intercontact micro wire recordings. And we hit what the choice of nuclei is based on the clinical picture. So and that depends on, so if there's a lot of midline or bulbar involvement, we tend to go for brainstem targets. And the brainstem targets we've been going for is substantia nigra reticulata and pedunculopontine nucleus. Now, at that depth, we can often be a millimeter one way or the other. So in some cases, I suspect we may be stimulating brachium conjunctivum, we may be in substantia nigra compacta, we may be in inferior regions of red nucleus. It's a little bit hard to know, and we're using the segmented leads so we can steer these things a little bit. But again, it is hard to know the structures. Also, many of these children, due to their initial injury, have some anatomic variation. So we don't, there will be change, you know, the atlases line up surprisingly well, but not perfectly. And the illustration here is based on standard atlases. So that may not be perfect either. But the idea is clinically, you try to get as many of the targets that we think would be relevant to that particular child. And again, it's also a risk-benefit analysis. While we have, in general, had very, very few complications, when we have, and the possibility of hemorrhage is definitely there, a brainstem hemorrhage is likely to have much higher consequences than hemorrhages in other areas. So that we will only do the brainstem targets in patients for whom we think that might actually have a life-saving effect. So there's the patient selection becomes very important, and the patient selection, target selection is very important. There's lots of surgical issues here, which Dr. Leiker and Dr. Elia and I and others have looked at, and still need to be further explored. But I guess hopefully that answers your question.
Yeah, it does. I was just wondering, it seems like that's not the case, but if the charge field could be big enough that could stimulate multiple of those nuclei with less electrodes, but it seems like they're.
I think that would be true. The problem is between all these spots, there's things you don't want to stimulate. So you get in trouble if you spread the contact, if you spread it too much, particularly brainstem. I mean, this is such a small area. So that and we're playing. So to give some sense, when we stimulate pedunculopontine nucleus, typical voltages are like 0.5 volt. When you stimulate in GPI, typical voltages are 3.5 to 4 volts. So GPI is a big area, and you can stay far away from things you don't want to stimulate, like external pallidum or capsule. So then we can just flood it and try to hit a lot of it. In PPN and in SNR, there's a lot of stuff there that you don't want to get anywhere near. And we can see, you can see eye movement abnormalities and things like that in that area, as you can imagine. So we have to keep the voltages low. We often keep the currents and the frequencies low in those areas. And we cycle things on and off. We use what's called a theta burst stimulation, so on for 100 milliseconds, off for 100 milliseconds, which seems to reduce some of the side effects. So there's both a temporal spread, because I think there's some areas that need continuous stimulation to be stimulated, and there's a spatial spread due to the voltage, now current mode, and pulse width. And so we need to control for both of those. But yeah, it's not good enough we can't just expand these areas in most parts of the brain.
I'll ask one more question, if that's okay. Our dystonia patient population is not the DYT1s, which is why I haven't been able to start that program here yet. But it seems like you're seeing, and I've seen sort of anecdotal stories of non-DYT1 responders. Can you comment a little bit more on that? I saw some pictures and videos, but do you have enough N to talk about responses to these non-DYT1s? And how about secondary dystonias like
so we do almost always non-DYT1. The DYT1 I showed was a rare example of a reoperation. DYT1, the targets, first of all, I don't think it matters that much. And anything in GPI will work. They're very easy to treat. And quite frankly, I send most of those kids to Phil Starr, who will just do this under MRT and asleep, and it's done, and the kids are home in a day. I see no reason to do this for DYT1 Parkinson's, essential tremor. If you know your targets, just go, right? But this is all in secondary, and that's the problem, which is in secondary, the distribution of injury is so broad and so different, and every child is different, that I think we have to do this. Almost all my kids are secondary. Now, what do you think of, what's the difference between primary and secondary? And I say, the typical example I use is something like glutaric aciduria. So glutaric aciduria type 1 has it's a genetic disorder. We know the gene, we know the metabolism, but it's a secondary injury because when you have one of the crises, the abnormal metabolites cause damage, and just as if you had an externally applied agent or a hypoxic injury. So I think in many cases, it's less about primary and secondary, and more about is the primary problem one of sort of inborn neuronal dysfunction, which is probably what DYT1 is, or is it a destructive process from any cause, whether it's a genetic or an acquired cause? But I think these destructive processes tend to be much more variable, often because there's different times in life when they occur, and because of environmental interaction, sort of the environment interaction with whatever the process is, which leads to variability in the children. We're getting about a 95% success rate, which means 95% of our kids are getting 5 points or more on the Berkefund Marsden. Most of them, our average is like 25 to 30 points on the Berkefund Marsden. Worldwide, the average for secondary is about 50%. So, and I just think it's real estate, location. You got to get the wire in the right spot, and it's not the same for everybody.
Yeah, I've got kind of an odd evolutionary question, if you will. I'm a retired neonatologist and been in a lot of the areas, but particularly more from biochemical point of view. The evolutionary question is that we've evolved biochemical systems that allow us to meet the energetics and structural functions of the brain and other tissues. And there's a lot of information about how evolution works in that biochemical context. We have evolved a common structure of brain. Homo sapiens has a brain that mostly looks like Homo sapiens. And that's been, there are a lot of interesting theories as to how that can evolve to preserve structure. What you're describing a little bit is an input-output function where the firing of the neurons is generating, as you put it, hash codes that are representative of the inputs that the brain has seen. And I wonder if you have any thoughts about how we've developed this information processing superimposed on the structural and biochemical functions such that we can all talk in the same room about subjects and understand each other. If each of us has a different function for interpreting a picture or grandmother picture, your analogy, how do we learn to do that in a way that we can pass on and stand in a room together and have a conversation if there's so much difference? Yeah, it's kind of a random question, but the evolution of this is really super information processing superimposed on other domains. And I'm just curious your thoughts of how that could have evolved.
So evolutionary theory is a morass and quite difficult to talk about. But of course, I think about this, and my very good friend Paul Chiswick at the University of Montreal has looked extensively at brain development and the computational components of brain development. But in terms of your question about how can we all interact with each other with brains that are wildly different from each other, one of the things I come back to, and I was saying this earlier this morning, we might have 30,000 or so genes in our bodies, and there are 100 billion neurons in our brains. So the genes can only be the assembly instructions. They can't do anything else. They can't tell you the content. You know, it tells you how to build the bookcase. It doesn't tell you what to put on it. So if you now have to evolve something where the assembly instructions are enough, where it's sufficient. So humans survive by group behavior, right? So the rewarded function is group behavior. So you have to have a genetic evolutionary process that leads to a computational structure which allows group behavior. And so what that essentially means is that the exterior component of this, our ability to communicate, our ability to interact, our ability to communicate emotions, has to be similar enough that despite the variations on the inside. Now, how does this happen? We learn in cultures and we learn from the people around us. So you build a learning box, but I think this learning box is interacting with the culture and the environment around it. And this goes to the fact that different cultures, in fact, do communicate very differently. The similarity of language between people is remarkable, and this is something that Chomsky and others have emphasized, although that may have less to do with built-in factors of humans than with the fact that language itself is maybe a much more unitary thing than we think. I think it's the power of the system that you can have wildly different internal representations and have remarkably similar behaviors from that. And if you think about it, that speeds up your learning a lot. In the field of computational neuroscience, one way to speed up your learning a lot is to have a network that's way, way, way too big, because now you can just load data into it, just like I could load to 10,000 images to it, right? Your brain's never going to fill up. Right? We have way more memory and processing power than we'd ever need. Simple calculations on the basal ganglia are that you could store or remember, in some sense, one image every millisecond for your entire life and still have used only a very small fraction of the storage abilities there. Right? You have this tremendous excess power. And what that excess power does is give you the ability to remember everything you have ever seen in your life instantly. It gives you very, very fast learning. It won't give you generalization. So you have other areas that try to synthesize this information and generalize to new things. So two different images of a tiger are still related. You understand they're a tiger, and the second one can eat you just as fast as the first one, right? So you can make these generalizations from that. But I think that there is real power to this over construction. There's real computational power to having more neurons than you could possibly need. And we clearly do, and lots of species, I don't know if elephants are smarter than us or not. They probably are, they're just not telling us, but they have, somewhere between 4 and 6 times as many neurons as we do in brains of similar structure, right? They just don't have opposable thumbs and larynxes. So I don't know. I mean, the evolutionary question is a very interesting question, but I think it is, what I would say is the brilliance of this is that you've figured out a universal computing machine that, when placed in any social environment, will adapt to that social environment. And it's in some ways the social environments that are the things that are evolving to give us the culture we have. That's coming straight out of Sapiens, but you understand where that would come from.